Okay, I think I'm going to go ahead and start. So for those of you who don't know me, I'm Allison Davis-Raboski, and I'm going to be introducing speakers for this second to last session before the coffee break. So it is my pleasure to introduce Dr. Ricardo Mallarino, who studies the genetics and development underlying phenotypic evolution in vertebrate animals. He got his bachelor's degree at the Universidad de los Andes, and then a PhD at Harvard University studying development of bird bills in Darwin's finches. He stayed at Harvard for a postdoc in Hopi Hoekstra's lab, looking at something that I think a lot about, which is the intersection between the mechanisms that produce color versus patterning in mammalian coloration. And he just this semester started as an assistant professor at Princeton, so please join me in welcoming him for his talk on how the mouse got its stripes.
Thank you, Allison. Thank you. It's really a pleasure to be here, and I want to thank all the organizers for the opportunity. I've enjoyed my time so far, and looking forward to the rest of the session. So I'll just get started right away because I don't have a lot of time. And I like to start with these images because every time I look at them, I just get amazed at this vast array of shapes and forms and colors that have evolved in nature. And I think as biologists, one of the challenges that we have is to try to understand how this great deal of biodiversity is originated. And by that I mean, what are the mechanisms that originate this? And so this can appear like a very general question, and it could appear quite daunting. So how do we approach it, right? That's one of the questions. How do we go about studying the mechanisms that generate this great deal of diversity? Well, if I were to show you these images from these embryos and then ask you to identify the group to which they belong to, I think you would probably be able to tell me that they correspond to vertebrate embryos, but I think you would have a hard time going beyond that. And the reason for that is because vertebrate embryos, particularly during their early stages, are very similar to each other. However, if I wait a few days, I collect these same samples, show you these pictures, and then ask you the same question, I think you would be able to tell me with much more confidence that this corresponds to an elephant, this one to a bat, this one is a turtle, you see the little shell forming here, this one is a frog, this one is a shark, and this one is a snake. And so the point that I want to make with this slide is that there are major changes in body patterning occurring during these developmental stages. And so I'm going to argue that if we really want to understand the factors that generate this diversity down here, we need to study what happens here in development. We need to study the effect that genes have during development. And so as a model to study this, I focus mainly on mammalian pigmentation patterns for many reasons. First of all, they're very diverse, as you can see as exemplified in these images here. Secondly, I think they're very interesting from a developmental biology perspective, which is where I'm coming from. They're very structured and very stereotypical, so that means that we can use them to address long-standing questions in the field of developmental biology, such as how does a tissue become organized in space and time. So in this perspective, I'm interested in understanding how do these pigment patterns form. In addition to this, I think they're also fascinating from an evolutionary biology perspective because they have evolved multiple times independently in many different groups. And so we can also use them to address long-standing questions in the field of evolutionary biology, such as do similar traits arise via shared genetic mechanisms or different ones? So on this other level, on the evolutionary biology level, I'm interested in understanding how do these patterns evolve. So there are many ways that we can study pigmentation patterns. Traditional models like the laboratory mouse have been instrumental for uncovering many of the genes and pathways involved in pigmentation, and that's in part or largely due to the wealth of molecular and genetic tools that are available, right? However, the problem with these mice is that their patterns for the most part are very simple. They tend to be monochromatic, and even if they were more complex, most of these strains have evolved in the laboratory, right? That means that they don't reflect the natural forces that occur in nature. On the other hand, we have more charismatic species like in this example, the giraffe, which have evolved complex pattern variation, but obviously with the downside that they constitute really bad models, right? So they don't have any molecular tools. It's not very practical to have a colony of giraffes in your lab. And so for my research, I try to sort of combine the best of both worlds. I try to study complex pattern variation that has evolved naturally in species that are accessible, but then I use some of the tools that have been developed in these model species to try to test some of these observations. And so the species that I'll be focusing in today is the African striped mouse, Rhabdomys pumilio. It's a species that has many interesting aspects about its biology, which I won't go into a lot of detail, but obviously the most salient feature, at least for me, is the fact that it has this very cool pattern of dorsal longitudinal stripes. And so we use this diagram to illustrate the pattern. So on either side of the midline, shown here in white, we have a dark stripe followed by a light stripe. Then there's a second dark stripe. Then there's this flank, which is sort of like grizzled in color. And then there's this very light ventrum. If we look very closely, we see that they have 3 different types of hair, or hair that can be classified into 3 different phenotypic categories. On the one hand, we see this hair that we call unpigmented. So no matter how close you look at this thing, you can't really detect any levels of pigment here. We've done all sorts of analysis with that. Then we have the second type, which we call black hair. It's full of this pigment called eumelanin, which I'll talk about in the next few slides. And then we have this banded hair, which is typical of a lot of vertebrate species, consisting of this yellow band of this pigment called pheomelanin, which is this yellow pigment that is, again, typical of a lot of vertebrates. And what's nice about this thing is that the way these guys make their stripes is by combining these different hair types in different proportions. And so what you're seeing here is the proportion of hair type along the different body regions. And so, for example, if you look at the dark stripe, it's mostly composed of this black hair, whereas the middle and the flank has sort of like an equal proportion of the 3 types. And then the light stripe and the ventrum have a disproportional amount of this unpigmented hair. And so then this is interesting because the question becomes, what gives rise to the spatial specific distribution of the different hair types? And so in order to address this, the first thing I wanted to do was to establish when, or to determine when this pattern is first established. And as I hope you can see here, by embryonic day 22, which is 2 days prior to birth, they have a gestation period of 24 days. You can already begin to see the pattern. At birth, the pattern is even more apparent. And by day 2 of development, or postnatal day 2, the pattern is fully established. So this is something that happens during embryogenesis. And so if we do histology from these early stages, we see something very quite interesting here, and is that hair follicles from the dark stripe have a lot of melanin deposited at the base, whereas hair follicles from the light stripe don't have that. So you're going to see a lot of these images. This is simply a sagittal section of a hair follicle, and here you see the base of the hair follicle, melanin here, no melanin here. So I wanted to characterize this phenotype a little bit better using different molecular markers. And so this is what I'll show you in the next few slides. But before I go on, let me just give you a brief overview of the way in which mammals produce their pigments. So mammals have these very specialized cell type called melanocytes. Melanocytes have to migrate and then differentiate in order to produce pigment. And so I'll spend some time talking about that because it's going to be relevant for the rest of the talk. They migrate from this specialized region called the neural crest. Then they make their way into the dermis, which is one of the layers of the skin. Then they migrate into the epidermis, which is the other layer of the skin. And then they slowly start migrating into the developing hair follicle. Then they sit at the base of the hair follicle when they produce pigment. As this process occurs, melanocytes start differentiating, and they express different markers associated with the different levels of differentiation. So I'll go over them because, again, they're going to be relevant for the rest of the talk. During early differentiation, they express SOX10 and KIT. Then they express this, what is considered to be sort of like the master regulator of melanocyte differentiation, this gene called MIDF. It's a gene that is absolutely required for melanocytes to differentiate into this sort of like star-like shape that is typical for neural crest-derived cells. And so here they express this enzyme called tyrosinase, which allows them to produce different types of pigment. And so depending on the amounts of tyrosinase, they can either produce black pigment, which we call eumelanin, or yellow pigment, which we call pheomelanin. When they have very high levels of tyrosinase, they go to this pigment. Low levels of pheomelanin, of tyrosinase, lead to the production of pheomelanin. And so we know from other studies that there are paracrine molecules that actually regulate the amounts of tyrosinase inside the cell. So these are molecules that are secreted from other cells, directly bind to the melanocytes, and then control the levels of this enzyme. So that's all you need to know about pigment pattern or pigment production in mammals. And so this is sort of like the canonical way in which mammals produce pigment. Okay, so what about our striped mice? So we asked a very simple question to begin with. We started by asking, can melanocytes actually migrate into the light and dark stripes? And I'll leave this diagram here so you can orient yourselves. And so what I'm showing you here is on the top row is a hair follicle from the dark stripe. This is stained for this marker here called KIT, early melanocyte marker. And next to it, I'm showing you the bright field image, just so that you know that I'm talking about hair follicles from the dark stripe and here from the light stripe in the bottom. And as you can see here, we have a very good amount of melanocytes in the dark stripe. We also see a good amount of melanocytes in the light stripe. And so here is what happens when you quantify this. So we actually see that there's no difference between the number of melanocytes found on either of the stripes, right? So this is a relevant result because it tells us that it's not a cell migration problem. Okay, so we know that it's not a cell migration problem, but what about differentiation? And this is where we see the first sort of like striking difference, right? When we use this marker that I told you was fundamental for controlling the terminal differentiation of melanocytes, we see that hair follicles from the dark stripe have a lot more cells expressing MIDF than hair follicles from the light stripe. And not only that, but also the intensity of expression is much higher. I quantified all this, and it's very robust difference and reproducible. And so this is relevant again because I told you that a melanocyte needs to be fully differentiated in order to produce pigment. So what's going on here is that these guys here cannot produce any pigment because they don't differentiate. And so I'm not going to bore you with these details, but we did a lot of things like quantifying the cell proliferation, cell death, hair follicle architecture, number. We don't see any of those differences. So this all suggests to differences at the level of transcription that are regulating some of these things. So in order to figure this out, so we asked the question, are there really differences in transcriptional activity? We took an RNA sequencing approach. Again, I won't go into much detail, but other to say that we use these samples from the different stages that I showed you before. I dissected out the dark stripes, the light stripes, and also the flank tissue to use as a control. We did RNA-seq. And so I'll walk you through what all this means. So this is what happens when we analyze all the stages together, and then when we group the samples by phenotype. So what you're seeing here is a comparison between genes in the dark stripe, shown here to the left, and genes in the light stripe, shown here to the right. This is expression level on the y-axis as a function of differential expression. So all the dots here that appear in blue are genes that are more highly expressed in the dark stripe than in the light. And then here, genes that appear or dots that appear in yellow are genes that are more highly expressed in the light stripe than in the dark stripe. So I want to point your attention all the way to the corner of this graph because we found a gene that was quite unusual. It's unusual because this is a transcription factor that has been mostly implicated in bone formation, never in pigmentation before. But nonetheless, it shows the highest difference in expression between the two regions. And then so we did a bunch of additional experiments, and we got excited about this gene because, as I told you, this represents all stages combined. But we were able to do stage-specific qPCR. And what we see here, when we look at the earliest time point, when we actually start seeing the pattern, this gene is already expressed at very high levels in the light stripe relative to the rest of the body. So this is exactly what you would expect from a gene that is involved in pattern formation, right? Early expression in the tissue that you think it's patterning. And so not only that, but we're also able to include tissue from the ventrum, which didn't make it into the RNA-seq project. And so we also see high levels of expression here. So again, we see this very interesting correlation between expression of ALX3 and the presence of this unpigmented hair. And so what I wanted to do next is to examine the expression of this gene during development in the developing skin. And so the approach that we took was to compare what happens in a non-striped species. So we use the laboratory mouse with what happens in a striped species. So we use our striped mice. So first I'll show you the data for the non-striped species. And so here what you're seeing are cross sections of mouse embryos. So you take a mouse and you section it like this from two different stages. So this is day 13.5, and then this is 2 days later. And this picture to the right is just a magnification of the picture to the left here. And so what you see here is that early on, you see expression in this region called the lateral mesenchyme. This expression then extends into the ventrum. And so you see it more up close here. And then when we do the same type of analysis in our striped mice in comparable stages, we see that we have a very similar expression pattern. We see initial expression in the lateral mesenchyme, which then extends into the ventrum. But then unlike these mice, in striped mice, we see this additional domain of expression restricted to only a few cells found here in the dorsum. So this is a picture where the neural tube is centered, and you see expression here, 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 here. So this is symmetric. It's very reproducible. So we were excited about this. It's very hard to know whether this actually corresponds to the future light stripe because the stripe only forms much later than this, but the location is very, very suggestive. And so later in development, when we actually see the phenotype and we do the same type of analysis, we see that ALX3 is expressed in many different cells in the hair follicle. And if we use a marker for melanocytes, we see that a lot of these cells correspond to melanocytes. So there is expressed in melanocytes, which are the cells, again, that will produce pigment. So to summarize this part, I've shown you that ALX3 is expressed in regions with light hair. It is expressed in the future stripe region, and it's expressed in melanocyte. So all this indicates that ALX3 might be somehow regulating pigment synthesis. And so obviously what you want to do is actually perturb the system in a way that can allow you to test whether this is actually happening or is it simply a correlation. And so in the next few slides, I'll describe the results from the gain-of-function experiments. I'll skip over the loss-of-function, but the results are largely complementary. And so for the gain-of-function experiments, we use these really cool technique called ultrasound-guided in utero lentiviral injections. So the way it works is that we come in with an ultrasound machine, and then we can target very specific regions of the developing mouse embryo, and then we can deliver a lentivirus, right? And so if you do it at the right place and at the right time, in this case, we do it at embryonic day 8.5, which is before the neural tube closes, so melanocytes are migrating, so you can infect developing melanocytes, which is exactly what we wanted to do. We want to test what is the effect of this gene in melanocytes. And so we obtain pretty robust levels of infection, as you can see here, and more importantly, we can infect melanocytes if we compare it to a melanocyte marker. And so we had two constructs, one carrying just GFP to use as a control, and then the other one which carried a copy of ALX3. And so, so the first question we wanted to address is ALX3 capable of interfering with melanocyte differentiation, right? This is what we think is going on. And so this is our diagram again right here. And so what you're seeing here is the control embryo is going to be on the top, and the experimental embryo is going to be on the bottom. So this is simply showing that we can infect a similar amount of cells under both conditions. So this is just GFP showing where the infection is. And then when we use this marker for differentiation, we see that control embryos have a lot of these cells expressing MIDF, and then we completely lose that in ALX3 infected embryos. This is a quantification of that. So we know that ALX3 is capable of decreasing the number of differentiated melanocytes. So then the next thing we wanted to figure out is, okay, is this something that, are we simply getting rid of all melanocytes, or is this something that is specifically affecting MIDF? And so the answer is, we're affecting MIDF because when we use SOX10, which is an earlier marker of differentiation, we see a good number of SOX-positive cells here, a good number of SOX-positive cells here. When we quantify that, we don't see that there's a difference. And so we know that this thing is specifically targeting MIDF. And so I'm just going to skip that part. And then, so the summary for the loss of gain of loss of function experiments indicates that ALX3 is decreasing, it's repressing MIDF, it's decreasing melanocyte differentiation, and it's repressing pigment synthesis. So the last sort of like component of this part was to figure out if this repression was direct or indirect. Is ALX3 directly binding to MIDF, or is it binding to other factors and interfering with MIDF downstream? And so, we use a bunch of experiments. I'll describe one of the assays that we did here. We know that ALX3 from other studies can bind to a sequence that looks like this. So we scan the promoter of rodents to look for these sites, and it turns out that it has 10 of these possibilities. And so we call these candidate binding sites. And to actually determine if they were actual active binding sites, we did CHIP qPCR. So in CHIP qPCR, basically what you do is you immunoprecipitate your melanocytes with an ALX3 antibody, and then you design primers around these regions, and then you ask whether you get a selective amplification of your target or not. And so by doing this, we were able to show that ALX3 indeed is binding to 3 of these sites in vivo, and so the interaction is direct. ALX3 is directly repressing MIDF. And so with this information in hand, we were able to put together a model for the way that stripes are patterned. So what happens in the light stripe is that we have a lot of this gene called ALX3, directly repressive MIDF, inhibits melanocyte differentiation, and then you get the production of this unpigmented hair. In the dark stripe, in contrast, you don't have ALX3. You have other factors that actually promote melanocyte differentiation, and then you have the production of this black hair. Sorry, I just want to make sure that I don't run over time. Okay, so so with this mechanism in hand, we wanted to determine, is this something that is unique to striped mice, or what happens if we go outside of this family, outside of this clade, and then we look at other striped rodents? And so this is the next thing that we asked, and what I'm showing you here is a phylogeny of rodents, family-level phylogeny of rodents. And in red arrows, I have indicated families that contain at least one species that has some kind of stripe pattern. So you see that it's evolved in at least 6 different groups. And so I already showed you what happens with the striped mouse, which sits here in the Myrridae family. And so I was able to collect skin biopsies from a mammal or from a rodent that is much more familiar to all of you, which is the chipmunk. And so this is a very, this rodent diverged over 70 million years ago from the Muridae family, but nonetheless, it has evolved a very similar stripe pattern, as you can see here and here. And so we asked the simple question, is ALX3 also involved in patterning these stripes? And so this is what happens with the striped mouse. We see strong expression of ALX3 in the light stripe and in the ventrum. And when we do the same type of analysis for the chipmunk, we see almost the exact same pattern, right? So this is, the stripes of these two rodents are being patterned by these same molecule, which was pretty remarkable. And so I want to go back to one of the slides that I showed you at the beginning and discuss some of the findings of this study in this context. So we started by asking how do pigment patterns form. I told you that the canonical mechanism for regulating color in mammals is by using this paracrine signal, so genes that bind to melanocytes and then regulate the production of these two pigments in a non-cell autonomous way. Well, here we find this new developmental mechanism whereby ALX3 is directly repressing melanocyte differentiation in a cell autonomous way, leading to the production of this unpigmented hair. So in a way, it's a way that evolution has to increase the potential for generating diversity of patterns, this new mechanism that was previously unrecognized. We also ask how do pigment patterns evolved. So I show you that this gene called ALX3 is normally expressed in the ventrum of all the rodents that we looked at, but in this species, it's expressed in a very specific domain here in the dorsal region. And so I also showed you that these two rodents, two species that have sort of a convergent pattern, but nonetheless have evolved, that have diverged a long time ago. They seem to be patterning the stripes using the same molecular mechanism. And so I think sort of like more generally, I think this study shows how we can combine sort of like transcriptomic approaches in non-model species with functional experiments in model species to go from organism down to tissue, down to the level of cell, all the way down to molecular mechanism. And I think more importantly, it shows how we can use development to understand biodiversity, but in turn use biodiversity to inform new principles in developmental biology. For example, this cell, the melanocyte, is a cell that is very important for humans as well. We didn't know it could be regulated in a cell autonomous way. And by looking at these sort of like non-traditional model species, now we know. And so I want to spend the last few minutes just talking about some of the things that I'm currently doing. Obviously this study answered many questions, but it also led to many others. So a large part of my lab is going to be focusing on some of these questions. And so I'll also discuss them in the context of these two questions that we've been posing all along. And so I told you that ALX3 is expressed at very high levels in the light stripe and at very low levels in the dark stripe, but we don't know why, right? And then, more importantly, we know that ALX3 is important for making a stripe lighter, but we don't know if it's important for making a stripe in the first place. And so I think we can ask questions such as what instructs the skin to form a stripe in the first place? And there's many different approaches that we can use. One of the things that I'm currently doing is we're trying to figure out what controls the spatial expression of ALX3. And so again, there are many ways of doing this. One of the approaches that we're taking is we're sort of assaying chromatin states at a genome-wide level. And so looking for things that are different in the light stripe than in the dark. So in this sort of like mock example, there's a region here that is activated in the light stripe that is not activated in the dark stripe. And so we're taking these regulatory sequences, we put them in front of reporters, and then we inject that into a lab mouse, and then we see if we can drive the formation of a stripe, which will tell us about the sufficiency of this gene to generate a stripe pattern. In addition to this, all of the stuff that I told you so far happens pretty late in development, so at embryonic day 22, which is 2 days before they're born. But we now have evidence indicating that the skin is patterned as early as embryonic day 14. So this is 8 days before all this happens. So we can go directly into these stages and then try to figure out the molecular identity of the stripe forming signal. And I'm happy to talk about all these things over coffee. And so, one of the components of the lab is to figure out the sort of like the genetic and biochemical way in which you make stripes, but then you also have to explain how do you generate variation in the stripes, right? We know that these stripe patterns show a lot of variation, so we already know, we have a pretty good idea of how this thing works in the African stripe mouse, but we know that pigmentation patterns vary in things like numbers, so the African zebra mouse has actually the entire dorsum covered in stripes, The chipmunk actually has the same pattern as these guys, but then the stripes are much more lateral, right? So they vary in position. And then they also vary in complexity. The 13-line ground squirrel, which is very common around here, has stripes in addition to spots. So we have to explain how these patterns also diversify. And so we can ask a very broad question, which is how is variation in stripe pattern generated? And to address this, we can take advantage of the fact that we have different species that differ in their stripe patterns that form natural hybrids in the wild. So this is one of the examples. This is a project that I'm actually collaborating with someone here in Michigan. We have the 13-line ground squirrel, which is here, and then the Mexican ground squirrel. And you see the intermediate hybrids that form here. So we can do a sort of like a genome-wide association type study and then try to identify the loci that control this variation. And then we can move into the lab, couple that with functional experiments in the laboratory mouse, and then ask the question, can we replicate some of these patterns that we see in nature? And so that was it for stripes. Sorry, I rushed a bit, but I was afraid I was not going to finish. And so, I think stripe patterns are a fantastic system, but I just see them as an entry point into understanding a much more general question, which is what are the molecular mechanisms patterning the mammalian skin? And mammalian skin, I think it's a fantastic system for many reasons, especially because it's very diverse. We have things like hair patterns, which I've talked about already. We have things like gliding membranes. This is actually a project that I'm collaborating with Bruce Ostro from Grand Valley State University. We're pretty excited about this because you also see convergent evolution in these patterns. You see sort of interesting gain of traits rather than loss, like a lot of people focus on. And so you have other things like spikes and scales. And so the idea is to use this as a model to understand the molecular basis for phenotypic diversity. And I think the skin is a pretty powerful system because it's diverse, it's accessible. We can do things like transplants, we can transduce it with a virus, we can grow it in a dish, so it's very accessible. Also because it's a very important tissue from a clinical standpoint. So we know a lot about the molecular mechanisms underlying the formation of skin. So that means that we don't have to start completely from scratch. We can use this as a starting framework to sort of understand how these pathways have been modified by evolution to generate this diversity. And so I'm going to end with a shameless advertisement. If you guys are interested in some of these questions, please come and chat. I just opened my lab like literally a month ago. And so I'm looking for people and interesting projects. And so with that, I'm going to thank my collaborators and funding agencies and you guys for listening.
Pardon me, I know you were looking at the stripes, but is your best understanding of white bellies that they have the melanocytes, they're just blocked maybe by ALX?
Yeah, that's a good. So we actually have a mechanism by which we know the mechanism by which white bellies are produced. It's a gene called agouti, which is one of the sort of usual suspects in pigmentation. And what happens there is that you have during development expression of agouti only in the ventral region, and so that it's a slightly different mechanism to what I showed here. But basically, it also interferes with melanocyte. Mostly pigment production because it acts directly on the melanocyte and it inhibits the production of pigment. So it's not a differentiation problem, but it does interfere with that. And it's all about the expression of this gene. And actually, work from my postdoc advisor, Hopi Hoekstra, has showed that if you go, you have two different populations of mice, ones that live close to the beach, others that live in the mainland. And then there's a shift in the expression of agouti. So the ones that live in the beach are whiter, or they have a larger region with white hair. And so if you look at the expression of agouti, it has shifted upwards, actually. So this is what's controlling the presence of the belly, of the white belly.
Hi, I came across your research when I was looking at popular science articles, and I was looking at the zebra stripe, the mice stripes on the back. Even within the same species, like if you were looking at a population of those same mice, do they have any variation in the thickness of the stripes on their back?
They don't, and that's something that I think about a lot, because if you think about, for example, zebra stripes or tiger stripes, they're used as fingerprints, right? You don't find two individuals that have the same pattern. These guys, you cannot tell apart two individuals from the stripe pattern. So they're remarkably consistent. So that tells you a lot about the underlying mechanisms regulating the spatial distribution of these stripes, right? It's really, really, really conserved, unlike other striped mammals. So yeah, you can't tell them apart. So if you go to a population of one of these species, all of them will look the same.
So my question is about the ALX3, right? So that's controlling the color, but not on its own. Like, what's controlling ALX3 expression patterning, right? Because that's what's dictating the stripes. Cis, trans, what do you know about the mechanisms?
That's the million-dollar question. We don't know. So this is why we're doing all these experiments now. We're doing ChIP-seq and ATAC-seq to try to figure out the upstream molecules, right? So ALX3 appears very early in development. So it could be that it's unleashing everything. It could be, you know, that it evolved an enhancer and it's responding to a positional signal that is present in all rodents, right? But then striped mice evolve an enhancer that actually activates ALX3. That's one possibility. The other possibility is that it's simply downstream of something that is activating the whole. So as I said, you know, if you go as early as E14, the skin is already patterned at that stage. So we're looking in those stages to see if we can identify sort of like the identity of this initial signal to make a stripe. But yeah, that's sort of like where we're going.
So across the phylogeny, there's groups that have stripes, don't have stripes. Has anyone looked at any sort of ecological associations with just the presence of striping pattern?
That's a good question. We're doing that. We're doing a little bit of that. What I can tell you is that in the African rodents, there's a clade that you have maybe like 15 species that have stripes, including these guys. And so all of those guys are diurnal, which is rare for rodents. Most rodents are nocturnal. So the transition of, you know, the shift to life during the day coincides with the evolution of stripes, which kind of makes sense, right? Well, like there's no point of running around at night, if you like with stripes at night, because this is thought to be anti-predatory adaptation. And so you know, you see shifts to open habitats, to diurnal behavior with the presence of stripes. I don't think anyone has really done you know, proper analysis to really figure this out, but it's sort of one of the things that we're thinking about. Okay, thank you.